So, hello students, welcome to Vedantu NEET English. This is Baswaraj sir, your biology master teacher. Quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know in the chat, all of you. Quickly let me know. Now students, let me start the class by a myth buster. Now what is a myth buster? All of us know, we humans, me and you, all of us, human, okay? We take in oxygen and we release CO2. Oxygen, CO2. But what about plants, students? What about plants? If you're in class 6th, 7th, 8th, 9th, even until 10th, you learn that plants take in CO2 and plants release oxygen. Students, is that entirely true? Let me tell you students, if you're in my class and if you're in class 11th, or if you love botany, let me tell you right now, it is a myth. Plants just don't produce oxygen, but plants also consume oxygen. So remember that, yes, plants consume CO2, but plants also consume oxygen. Now students, let me ask you one more thing. Like when I consume oxygen, right? When I consume oxygen, respiratory system is involved. I have lungs. Yes, I have lungs. With the help of lungs, I can breathe. But do you think plants also have lungs? Quickly tell me in the chat, students. Quickly tell me in the chat. Do you think plants also have lungs? All of you? Do you think that? Yes, plant is like behind your back. It is like fixing one lung, one left lung, right lung. Is it true? No. So if plants do not have lungs, how can they do breathe? Like how can they even breathe at all? If they do breathe, that is. Now that comes to one more question. Okay. What is the difference between breathing and respiration? How many of you know that? Raise your hands. Right? How many of you know that? Raise your hands that if you know the difference between breathing and respiration. Let me tell you. Okay. First of all, the process. Breathing is a more of a physical process, while respiration is more of chemical process. This is physical process. It is a physical process and chemical process. The next thing is purpose. The purpose of breathing is to take in oxygen and release our CO2 in us. Now, what is purpose of respiration? The purpose of respiration is to produce energy. Okay. O2 and CO2. Now, do we have control over breathing? Yes, we can control breathing to some extent. But can we control respiration? Absolutely not. We cannot control respiration. Do we produce energy in breathing? No. No energy is produced in breathing. At the same time, respiration energy is produced. Now a question arises to all of you. Breathing, all of you are aware from the chapter respiration, right? Now, where do you think respiration takes place? Like what is the exact location of respiration? If I ask that question, tell me how many of you tell the answer. Tell me now. Where do you think exactly? In what part of the body do you think respiration takes place? Is it happening in the eyes? Is it happening in my hair? Is it happening in my hand? Or is it happening in my heart? Tell me students, where is the respiration happening? Let me tell you again. Respiration is taking place inside what, students? Respiration is taking place where? Respiration is taking place inside a cell. If you look at a typical cell, like the students, if you look at a typical cell, inside the cell if you go, inside the cell if you go, you will find many organelles. But where exactly happens inside the cell? Where does it happen? There are two places. Respiration mainly starting point of respiration is happening in the cytoplasm. It is happening in the wide cytoplasm. But if I ask you, where is the oxidative decarboxylation? Where is Krebs cycle? Where is ETC happening? You will tell me, sir, all of these two reactions, Krebs cycle as well as ETC, that is taking place under a separate type of organelle right here called as mitochondria. Now, where exactly inside mitochondria, right? And what is the inner outer? That I will teach you again later on with the help of animation. So with that basic information, respiration occurs inside cytoplasm and mitochondria. With that basic information, can we start today's class? Can we start, students? Can we start today's amazing class of one short lecture? one short lecture on respiration in plants. If yes, if you are interested for this class, quickly smash that like button and also subscribe to the channel if you are new. And also everyone, green hearts in the chat for the bot army. Go, all of you. Now, let me tell you one complex definition of respiration first. Then I'll make it easy for you. Okay. One complex definition, I'll tell you what is the meaning of respiration. Okay. Respiration is basically biological. It is basically biological oxidation of organic molecules. Very fancy, no? Very fancy. Biological oxidation of organic molecules. Breaking of CC bond. The food is getting, the substrate is getting converted to energy. All that is very complex. But here I am to make it easy for you. Now, all of us saw a cell, right? All of us saw a simple cell. Let me draw the cell in the board for you. Let me draw the same cell on the board for you and make the entire concept a very easy way to approach this. For example, student, this is a plant cell. Let's take a plant cell with a cell wall. Okay, this is a cell. Let's take this as a cell now. Now, into this cell, what is entering, students? Plants require oxygen for oxidation. Oxygen is entering into a cell. I'm going to make it easy for you. Listen to me. Oxygen is entering inside a cell. Now, in the same time, inside a cell, there is one more substance called as food. This food is basically glucose. Okay. Now, this oxygen is acting on the food and it is undergoing a process called as oxidation. It is undergoing a process called as what? Oxidation. So, the food is undergoing oxidation. Now, what happens in oxidation? All that you will learn in this class itself. Okay. So, listen to me. Inside a cell, oxygen is acting on the food. The food is undergoing oxidation, and when the food undergoes oxidation, what happens? The food is basically carbon based. This, whenever it undergoes oxidation, there is breaking of carbon-carbon bond. Breaking of carbon-carbon bond of the carbon-based molecule, glucose. And when this bond is broken, energy is released. And this energy is stored. This energy is stored in the form of ATP. That is all is respiration. That's all you need to know. So what is respiration, students? Respiration is a biological, inside a cell, oxidation of organic molecules such as food, glucose. When it undergoes oxidation, energy is produced and the energy is stored in the form of adenosine triphosphate. That is the basics of respiration you need to know before you understand today's class. Now, what is oxidation? What are the steps in oxidation? What is glycolysis? What is Krebs cycle? What is electron transport system? All that you will learn in today's class. Okay, let's go to the next concept, students. Now, here I spoke about food, right? I spoke about food. The food I spoke about. The food which I spoke about is nothing but the respiratory substrate. Fancy name, simple word is food. Now, respiratory substrates, students, can be of variety types. Now, quickly everyone in the chat tell me, what is your favorite biscuit? See, I love biscuits. I love eating biscuits. I grew up my entire childhood by munching on biscuits like there is no tomorrow. Tell me in the chat, students, what is your favorite biscuit in the chat quickly? Similar to you, how you have a favorite biscuit, similarly, a cell also prefers, a typical cell also prefers a special type of food. The food which I'm talking about is not a biscuit here. The food I'm talking about is carbohydrate. It is carbohydrates. So carbohydrates are a typical, like it is a typical food which our body loves, a cell loves. Okay, carbohydrates. Apart from carbohydrates, our body can also take in the fats, or can also take up the proteins and also certain organic acids. So all of these substances can be broken down with the help of our body. Okay. Now, apart from this, students, apart from this, there are other substances such as, let's say, water. Let's talk about hormones, minerals. Students, do you think our body and the plants, can we use minerals, hormones and water as a source of energy? Yes or no? Tell me. Yes or no? No. It cannot. This is no. This is yes. So plants are using, or our body is using generally, in general term, carbohydrates, fats, proteins and organic acids like malic acid and acetic acid and everything can be used as a respiratory substrates, while water, hormones and minerals cannot be used as a substances. Clear? Clear? All of you? Yes. Now, before we go ahead, students, all of you should be aware of a simple process called as metabolism. Metabolism is divided into two separate categories. One is catabolism, other one is anabolism. Now, students, from a complex substance, I am making simple substance. What do you call that? From a complex substance, I am making a simple substance. Can I call it as a catabolism? Catabolism. So, for now you remember, the entire process of respiration is catabolism, but it is not true. Why, I will tell you at the end of the lecture. So watch the video till the very end if you want to know the answer why it is not catabolism, or it is not even anabolism also. New term I'll teach you today. Okay. Apart from that, students, in this entire reaction, energy is released. So can I call it exothermic reaction or endothermic reaction? Tell me in the chat quickly. I will call it as exothermic reaction. Okay, exothermic reaction. Clear, all of you? Clear? It is also a physiochemical reaction. There is physical phenomena, that is exchange of gases, and chemical phenomena, both are happening in the respiration. And students, if you want these notes, right, all the notes are available on the Telegram channel, Respiratory Biology Telegram channel, you'll get it. And also, students, do you want DPP today? Like a surprise from my end, before we end the year, do you want DPP today itself? What say? So after today's class, if you go to the Telegram channel, you will get around, let's take 40 questions as DPP today. Have fun. So before the year ends, keep this chapter, all of you have backlogs, will be clear today only. Okay, so solve these questions today itself. Do not wait. So go to the channel today and solve the DPP today itself. Okay. Now. Students, can anyone of you identify what is the structure on the board? A celebrity structure here. A celebrity on the board. Can anyone tell me who is the celebrity on the board now? All of you. Who is the celebrity? I have seen this somewhere, no? Like it has some 3-phosphate here. 1, 2, 3 triphosphate. Who is the celebrity? This celebrity is none other than ATP. Okay. This celebrity is none other than the ATP, adenosine triphosphate. Now, why is ATP important? Let me give a small instance. Okay. Now, students, how does the money come to your house? Simple question. How does the money come to your house? Quickly tell me in the chat, students. Quickly tell me in the chat. For example, a mother or father or someone is earning. The money is coming to your house. Yes or no? Money is coming to your house. It may be coming weekly, monthly, yearly, or if you're very rich, daily. Okay. So it will be coming some way or the other. The money is coming to your house, and with the help of that money, you are able to afford the lifestyle, or if you're able to buy whatever items you desire. For example, if you want a Lamborghini, go buy a Lamborghini. If you want a medical seat, you can buy a medical seat. Yes, you can. So, my students, if you do not have much of money, don't worry. Medical seat is also very, very cheap also. This class will help you there. Okay. Now, apart from that, you can buy everything. So, like how you can use money to buy everything, right? You can use money to buy everything. Similarly, inside a cell, ATP can be used to make anything. All of you understood? Like, in the world, you can use money to buy so many things, whatever you desire. Similarly, in the body, we can use ATP to make or do something. So that is why, that is the main reason. In every single organism, energy currency energy currency through which we can do certain phenomena or do different types of reactions is possible because of ATP. So ATP is like the money which you have in real world. Now students, how many of you know how to write check? How many of you know how to write check? Okay students, let me tell you a small hack, okay? Life hack. Someday all of you will write a first check. Someday all of you will write a first check. No. Let me tell you, please be careful, because the insane amount of pressure you will have on you is going to be very large. So practice beforehand how to write a check. Okay. Why did I tell about check right now? Listen to me. Now, this is a check. Now what do? Can you use the check and give this check to a vegetable person and like, give me 1 kg potato, tomato? The person will throw the check at your face. You're like, what is this check? Yes or no? If you take this check and you take the check and go to vegetable seller and give him this check, and if you ask him, Anna, 1 kg potato, check one, he'll give back. Okay, he'll check it on your face. So you can't use the check directly. So what do? You take the check, you go to the bank, deposit the check. As soon as you deposit the check, you get the money in return. All of you are aware of that. What is check? I can't explain. How will I explain what is check now? Okay, listen to me now. So if you take the check, deposit in the bank, money will be given to you. Then you can use the money to buy the groceries or buy the potato, tomato, anything. Similarly, how check can give you money. In the case of our body, there are certain molecules called as NADH and FADH. Just like a check, NADH and FADH can produce the currency. Like how a check can produce money, NADH and FADH can produce ATP. It can produce ATP. 1 NADH, 3 ATP, 1 FADH, 2 ATP. According to your NCERT, which is not true actually, but you have to believe NCERT only, no. If I tell you the actual thing, if you write and come, you will not get marks, you will come back to me. Sir, you told 1.5, 2.5, exam they give 3 and 2. So, whatever the real concept is, forget for now, follow NCERT. 3 ATP and 2 ATP. Remember that. Safe aside, no, safe aside. Okay, safe aside. Now, this is the concept of check and money. Now, students, how can we use it? Like, how do we use the ATP and how is the ATP formed inside a cell? That is very important to understand. For example, see this. This is a adenosine diphosphate. Di 2 phosphate here. To this adenosine diphosphate, I am adding this one more phosphate. Now I cannot simply add it. Can you get the money for free in this world? Not true. Not true. No, not true, student. Not true. Cannot get. There is no easy way. You have to put in the energy to get the money. Similarly, if you want the ATP, you need to add the what? Like how you cannot get money in the world, in the real world without energy, you cannot get ATP without energy. So if you want ATP, adenosine triphosphate, to this ADP, we need to add energy, and when we add the energy, this phosphate will finally bind here. This phosphate will bind here. So if you want to convert ADP into ATP, ADP into ATP, what needs to be added? Energy needs to be added. Only when energy is added, ADP becomes ATP. And let me tell you, all the energy is stored in the bonds. Like James Bond, all the energy is stored in the bonds. Now, with that concept in mind, now I have a lot of ATP, lot of money. Now I want to do something in life. I want to spend that money. How can I spend? Now, if you have a lot of ATP, how do we spend this ATP? That is also very important. All I told you, all the energy is stored in the bond. All of you agreed, all the energy is stored in the bond. What if I break the bond? Friendship bond can be broken, like just like how friendship bond can be broken. Anything can be broken here. So this ATP has adenosine triphosphate. If I break the bond open, phosphate is released. Energy is also released. Simple. So we have adenosine triphosphate here. Now if I break this bond right here, phosphate is out, energy is out. Use the energy then. Use the energy. Simple. Does it get more easy than this? That's all. So if you want to make ADP to ATP, add energy. If you want ATP to ADP, again back to ADP, you have to break bond. When the bond is broken, energy is released out. Energy is released out. Okay. Now students, till now, any doubt anyone? Till now students, any doubt anyone? Sir, use the same PDF. Yes, obviously. I can use the NCERT lines or I can use the same PDF. What will I do? Huh? Now tell me students, till now any doubt anyone? Any doubt anyone students? Quickly tell me in the chat. Any doubt anyone till now? Any doubt anyone? Very simple, right? Till now the chapter is very simple, very smooth. So whatever am I teaching you right now? Is the basics to this chapter. First, what did we study? We studied right now, let me give a small recap. We learned what is breathing versus respiration. We learned what is respiration. We also learned what is respiratory substrates. We learned that how ATP becomes an energy currency. Now with that in mind now students, let's understand what is anaerobic respiration and what is aerobic respiration. Small difference, major impact. Okay. Now first of all, what is the meaning of anaerobic respiration? The meaning of anaerobic respiration, it means it occurs in the absence of oxygen. Oxygen is completely missing here. Gone case. Oxygen is missing. Now where does it takes place? It takes place in the cytoplasm. Okay. Now what are the products here? The products are going to be ethanol. If you know the answer, type in the chat. Ethanol plus ethanol, or it can be lactic acid. Can be here. And also students, the energy produced here is very less. Less energy is produced here. At the same time, aerobic respiration, it occurs in the presence of oxygen. Where does it takes place? It takes place in the case of cytoplasm and mitochondria, as well as mitochondria. Okay, mitochondria. Next important point here, ethanol plus lactic acid. What do you think we get here, students? Here we get CO2 plus H2O are the byproducts here. Apart from that, students, here more energy is liberated. More energy is produced here. Okay, more energy is produced in the case of aerobic respiration. Okay, very simple thing. We'll go into detail of it more and more. Okay, now this is a type of respiration, aerobic respiration. All of you know, glucose plus oxygen, food plus oxygen, energy is being released here. Which we take in, right? That is the energy we need. Carbon dioxide plus water is the byproducts. This is the entire aerobic respiration. Now students, understand one small concept here. I'll ask you a simple question. Can you finish your entire NEET syllabus in one day? Is it possible? Even if you try thinking about it? Is it possible to finish the entire NEET syllabus in just one day if you sit physics, chemistry, biology, botany and zoology? Not possible at all. So how do you do your preparation? You take your time, you divide the entire workload into different sections, and little by little, step by step, you finish your syllabus. Yes or no? Sometimes you can have backlogs, you clear them eventually. Step by step, you finish your syllabus. Similarly, in the case of a cell, energy cannot be stored in one shot. The energy which is produced by the process of respiration, by the process of oxidation, cannot be stored in one step. So what do? We have multiple different steps by which the energy is finally stored in the form of ATP in multiple steps. Now students, right now I'll give you an entire overview of the chapter. Ready? First of all, let's take a food. Glucose. Now this is in the case of aerobic respiration, glucose. Glucose in the first step gets converted to a substance called as pyruvate. Pyruvate, okay, pyruvate, students. Now students, tell me, this glucose to pyruvate, this step is called as glycolysis. It's called as glycolysis. Now pyruvate gets converted. Pyruvate eventually gets converted to acetyl-CoA. Acetyl-CoA. This step is called as link reaction, gateway reaction, oxide decarboxylation, whatever you call it. Link reaction. Link reaction. And this acetyl-CoA eventually, enters the Krebs cycle. Eventually enters the Krebs cycle. Okay. Now, this Krebs cycle, products such as NADH, FADH, all the NADH and FADH, they enter a last step called as electron transport chain, or you can also call it as electron transport system. In this step right here, all the energy is used to produce the ATP. This is the entire chapter which you have to learn today itself. Okay, and this one by one, step by step, the entire reaction goes on. And let me tell you, this step right here, glycolysis itself has 10 steps. Link reaction, a single step. Krebs cycle, it is an entire cycle. Then we have ETC. ETC also has 5 different protein complexes. Here we have 5 protein complexes. So don't you think so? This is like a roadmap to your NEET journey, you know. This is like a roadmap to your NEET journey, like how we have roadmap, right? How we have a roadmap to your NEET journey and everything. This is called as a roadmap for ATP, and that is your entire chapter. Now students, tell me honestly, how many of you find it difficult to understand glycolysis, Krebs cycle, etc? Honestly, raise your hands. Raise your hands quickly. Don't be shy, raise your hands. Okay. Tell me students, how many of you find it difficult to understand any of the following? Raise your hands. Because if you do so, I promise you, it's a deal from my side to your side. If you watch the lecture till the very end, you will get the best clarity on all of these topics. And when I say clarity, I mean I'm going to make you understand, not make you by heart any of it. That is my deal, and this is how I've always been teaching this chapter. Like every single time you learn the glycolysis from me, you will actually understand glycolysis, not just by heart it. Okay, that is my deal from your side. Okay. Okay. Can we start? The first question to all of you. Students. Now I want everyone to be very active. I want everyone to be very energetic. In this process, it's a two-way thing, right? So if you participate, answer the questions, you will learn more, you will get more and more energy. You'll get more information also. Okay. The question is, where do plants get the respiratory substrates from? Like how I am eating food and I am drinking water. I am eating biscuit, anna sambar, curd rice and everything to get my energy. Where do you think plants get the energy from, get the respiratory substrate, that is the food from? Food. Like, for example, do you think in the night time they're hiding behind and eating the food in the dark? There's something called dark reaction or light or dark reaction or happening. Students, do you think so? The answer is right here. If you remember the previous chapter, that is process of photosynthesis, in this process of photosynthesis, there was a production of glucose. How many of you remember that? This glucose right here is actually used by the plants for the respiration. So plants are only preparing the food, plants are also using the food to produce energy. Truly wonderful, no? Like we have to eat this from outside. We have to get this oxygen also from the outside. But plants are only preparing glucose, plants are only producing oxygen also. Truly self-sufficient. Truly self-sufficient, students, truly self-sufficient. Okay, next question. Do plants breathe? Next question to all of you. Do plants breathe? Do you find them breathing? The answer is not very technical. That answer is very controversial because they don't really breathe, but they need oxygen. Like for example, how we breathe through our nose and exhale out through our lungs and mouth and nose and everything. Plants do not have any separate organ at all. They do not have any special organ at all. Rather, they don't require any organ. Let me tell you why also. But before that, there are certain organelles in plants which help in exchange of gases. For example, a stomata. A stomata can help in exchange of CO2 and oxygen, right? A help in that. In terms of woody plants, there is something called as lenticels on the woody plants which can help in exchange of gases again. Exchange of gases. But apart from stomata as well as lenticels, apart from stomata and lenticels, plants do not require, I repeat again, plants do not require special organ for breathing, okay, for breathing. The first one is very simple, understand. That is, each plant part, the first point here, each plant part can take care of its needs. Each plant part can take care of its own oxygen needs, it can take care. Even though it can take care of its own requirements, let me tell you students, they do not even require so much of oxygen. Not required at all. Root, stem as well as leaf, they do not require a lot of oxygen at all. So the second point is demand for oxygen is very less. Even if there is some demand, it can be achieved. It can be achieved by photosynthetic byproduct. Photosynthetic byproduct, that is oxygen. The O2 released because of the photosynthetic byproduct, oxygen, the same oxygen can be used by the plant to take care of its need. And the third point is even more simple to understand. If you look at a typical stem, students, the inner part is mostly dead. Mechanical tissues for support and everything. Most of the living cells, most of the living tissues are mostly in the outer surface here. Most of the living tissues are on the outer surface. And every single time oxygen enters with the help of lenticels and everything, the diffusion can be very easy because all the organelles are near the surface. So diffusion can be very simple and they do not require a special organ. Diffusion can be very easy because all the living tissues are present in the periphery or the outer layer. So easily oxygen can diffuse. So they don't require a special organ per se. Okay, do not require at all. Okay. Now students, one small concept before we go to the main highlight of today's class. Are there any similarities between combustion and respiration? Yes, there is. Because in both of these reactions, there is breakdown of complex substance, production of energy and using oxygen. When you try to burn something, it needs oxygen, it produces energy and complex substances broken down. That can happen. But obviously there are differences. I will give you the notes. You can read the differences. Not very important. Now we come to the main highlight of today's class. That is the first step in glycolysis. Can we start now, students? Can we start? The first step in respiration, technically, not glycolysis, first step in respiration. Can we start? All of you? Can we start? All of you quickly smash the like button. All of you quickly smash the like button. And can we start the first step in all types of respiration? It could be aerobic, anaerobic, prokaryote, eukaryote, unicellular, multicellular, every single type of organism. This is the first step in respiration. Can we start? Now I want everyone to be very, very, very attentive right now. Very, what do I say? Always maximum effort right now and ultimate attention. I do not want you to look around here and there for the next 15 minutes. 15 minutes is going to change your life the way you look at glycolysis. After 15 minutes, you can chill a little. Can we start now? Can we start? 15 minutes, time starts now. Keep counting. Now students, what is the meaning of glycolysis? Glycolysis is the first step in respiration, all type of organisms. That point is clear to all of you. The next point is, what is the meaning of glycolysis? Glyco means sugar. Glyco means sugar and lysis means breaking. So what exactly is happening here, students? In the case of glycolysis, there is breakdown of glucose molecule or breakdown of sugar molecule. Okay. Now the first main point which everyone needs to understand is the location. It is the location of glycolysis. Location of glycolysis is what is the students? Tell me in the chat quickly. Location of glycolysis is the cytoplasm. It is the cytoplasm. Next question, next thing is number of steps. The number of steps involved. There are totally 10 steps here, students. 10 steps. We will learn all of them. 10 steps. Next thing is what exactly happens here, right? That is, plants generally produce glucose. This glucose is stored in the form of sucrose, right? Stored in the form of sucrose. Now, if a plant has to do glycolysis, it needs to convert this glucose, convert this sucrose back to glucose. Plants prepare sugar, that is glucose, stored in the form of sucrose. Before the glycolysis start, plants has to convert the sucrose into glucose with the help of enzyme called as invertase. Invertase enzyme. Okay, invertase enzyme. Apart from this, this reaction occurs in aerobes as well as anaerobes. Now students, you tell me in the chat, all of you, if this reaction is taking place in anaerobes, does this reaction require oxygen? Does the first step in respiration, does it require oxygen? No. This step does not require oxygen. O2 is not required at all here. O2 is not required. And this reaction, I told you, this glycolysis takes place in the case of prokaryotes, eukaryotes, unicellular, multicellular. Every single place it takes place. Remember that. Okay. Now, this entire glycolysis is also called as EMP pathway. It is called as EMP pathway. Why? Because this glycolysis pathway was discovered by three main scientists called as Emden, Payoff and Parnas. EMP. Emden, Payoff and Parnas. Three different scientists. This first letter of second, EMP pathway, it's called as glycolysis. Okay. Now before we start glycolysis, there are two things which you need to understand. What is the meaning of oxidation and what is the meaning of reduction? Basic concept, back of your head you should know. What is oxidation? Well, oxidation is basically loss of electrons or loss of protons. What is reduction? Reduction is gain of electrons and gain of protons. Is that point clear to all of you? What is oxidation? Loss. Reduction, gain. Please remember this for the entirety of today's class. Very important concept. Now, the entire glycolysis can be divided into two separate steps. One is called as preparatory step, other one is called as payoff step, right? Before you earn a lot of money, you need to invest some money into a business. For example, you're trying to do a business. For example, let's take someone's name, let's take Preeti, okay? Preeti is trying to make one restaurant. First, she has to invest the money in the restaurant, buy some cookware, get some ingredients, and then when the restaurant is ready, she'll get the money back by the customers. Similarly, if you want to, in the entire glycolysis, the 10-step process, first is called as preparatory stage, and the second stage is called as payoff stage. You invest first, then get the returns. Okay. Now, first step. Listen to me, all of you, very carefully now. First step. What exactly is called as the first step? Here. On the right side is your NCERT diagram. Here is the first step. In the first step, there is a process called as phosphorylation. It is called as phosphorylation. In phosphorylation, there is addition of phosphate. Okay. For example, this is a glucose molecule. How many carbon? 6 carbon. 6 carbon glucose molecule, and this is a ATP. Now students, if I have to add this phosphate to here, the first thing which I need to do is break the bond. When I break the bond, this phosphate is free now. So glucose, if I take this phosphate and I add it to the, sixth position, you tell me what happens? Glucose will become glucose 6-phosphate. Yes or no? All of you tell me in the chat. See here. Glucose to glucose 6-phosphate. 1 ATP is consumed. So we are using 1 ATP and converting the glucose into glucose 6-phosphate. And ADP becomes ADP with a loss of 1 phosphate. That is the first step. Glucose became glucose 6-phosphate, and remember students, you need to know the first enzyme and the last enzyme. That's all you need to care about. First and last enzymes. Here the first enzyme is hexokinase in the phosphorylation. So glucose became glucose 6-phosphate. Now, second step is called as isomerization. Now what is isomerization? Same but different. What do I mean by that? If you know basic chemistry, you will tell me what is isomer, right? What is an isomer? If you know basic chemistry, you can tell isomers are molecules with same chemical formula, C6H11O9P, same chemical formula, but structurally they are different, right? Different structural formula. So chemically they are same, structurally they are different from each other. Now the second step, here, if you notice the NCERT, first step, second step. In the second step, glucose and fructose are isomers. So they're changing the shape. See here. So glucose is becoming what? Glucose 6-phosphate is isomerized into fructose 6-phosphate. Structure different, same chemical formula. So in the second step, first step, second step, fructose 6-phosphate. Now, what is the third step? In the third step, again we are adding a phosphate. Phosphorylation, third step also. Okay, so this is fructose 6-phosphate. Here I have an ATP. Now I'm breaking the bond. Let's say I break this bond right here. Break the bond. Now I take this phosphate and I attach to the first position. Now, can I tell students, if I add the phosphate to the first position here, fructose 6-phosphate, can I now call it fructose 1,6-phosphate? All of you agree with me? Right? So fructose 6-phosphate, because of addition of 1 phosphate from ATP, which is getting converted to ADP, obviously, it is getting converted to fructose 1,6 bisphosphate now. 1,6 bisphosphate. Done. Fructose 1,6 bisphosphate. Done. That till here is the entire preparatory stage. That is all. we learned that one more step is there. Okay, preparatory. Glucose to glucose 6 phosphate, addition of one phosphate from ATP. One ATP is used here, first step. Second step is called as isomerization. Isomerization done. Okay, isomerization done. 1 second, students. Okay, isomerization done here. And then what is happening? One more ATP is used to add one more phosphate. So fructose 6 phosphate to fructose 1,6 bisphosphate. Till here, all of you are clear. It is clear. Now understand what happens in the fifth step. Okay, see here. We are done till here now. Done till here. Fructose 1,6 bisphosphate. Now students, understand. This fructose is how many carbon? 1, 2, 3, 4, 5, 6. The fructose right now is 6 carbon molecule. It is a 6 carbon molecule. Now what exactly is happening here is, this 6 carbon molecule is actually breaking into half. Breaking into half. And this will lead to a 3 carbon molecule this side and a 3 carbon molecule this side. That is what is happening here. Can you see, all of you? In the next step, that is first, second, third and fourth step. In the fourth step, we are getting gap here, GAP, that is glyceraldehyde 3-phosphate, as well as DHAP, dihydroxyacetone phosphate. So what is happening here is, fructose 1,6 phosphate, 6 carbon is breaking into 3 carbon and 3 carbon. See here. Into 3 carbon molecule of glyceraldehyde 3-phosphate and 3 carbon molecule of DHAP, that is dihydroxyacetone phosphate, 3 carbon, 3 carbon. But one more interesting part here now. Interesting thing is, this DHAP and GAP both are isomers. So if both are isomers, can they interchange now? Yes, can they interchange? Yes, they can. So this DHAP will convert into glyceraldehyde 3-phosphate, and that is why from this step onwards, instead of writing two separate things, since DHAP, dihydroxyacetone phosphate, is converted to GAP, now onwards I will write 2 molecules of GAP. Instead of 1, I will write 2 now, or PGAL, whatever you call it, PGAL, phosphoglyceraldehyde. See, students, there are 3 names here. It can be called as GAP, or it can be called as PGAL, glyceraldehyde 3-phosphate, phosphoglyceraldehyde. Okay, phosphoglyceraldehyde. Okay, or it can be called as triose phosphate also. Now from here onwards, since we have converted dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, from here onwards, everywhere you can see into 2, into 2, into 2 and into 2, every single place. Instead of writing 2 separately, we are adding it into only 1 now. Now that is the fifth step. Isomerization is the fifth step. First, second, third, fourth, fifth. Now what is happening in the sixth step? From sixth step onwards, what starts? Payoff stage begins now. Ready, all of you? Ready to get all the money back into the account now? Yes, we are ready to get all the money back into the account. Listen to me very carefully what happens in the sixth step. From after isomerization, in the sixth step, there's a process called as oxidation is happening. Now students, if you remember, quickly tell me in the chat, what was oxidation? What was oxidation? Tell me. Oxidation is loss of electrons or loss of protons. Do you remember that? All of you remember? See here, what is oxidation is happening here? So when oxidation takes place, electrons are lost, protons are lost, energy is also produced. It is produced. Energy is produced here because of oxidation. Now listen to me very carefully. This electron and this proton, they are called as redox equivalents. They are the products of oxidation. GAP, glyceraldehyde 3-phosphate, is undergoing oxidation. This electron and this proton are taken up by a special molecule right now called as NAD+. they are taken up by NAD+. So these electrons and protons are taken up by NAD+. Now students, tell me, what do you call gain of electrons on gain of protons? What do you call that? See, loss of electrons and loss of proton is oxidation. Oxidation is loss. What do you call gain? Gain is called as reduction. So NAD+ is taking the protons and electron and is undergoing reduction to produce NADH. And that is how students, that is how in the 6th step of your glycolysis we end up getting a NADH. Now what is the use of this energy? Listen to me very carefully now. This energy right now is used. This energy is used to bring. Energy is used to bring inorganic phosphate. Inorganic phosphate from the cytoplasm. From the cytoplasm. And this energy is used to form a bond here. So what happens? Magic. So in the sixth step, very complex, I know, but if you're watching my lecture for the first time, you'll be like, wow, does it really happen like this? It does. Instead of making you by heart the 6th step, I made you understand right now how this NADH is produced. And if you're watching this lecture for the very first time, I would recommend watch the lecture one more time in a 2x speed to understand the concepts better. Okay. So in the 6th step, what is happening? In the 6th step, glyceraldehyde 3-phosphate, 2 molecules of glyceraldehyde 3-phosphate is now converted to 2 molecules of 1,3. So here we had only 3rd position. This phosphate is added, right? It became 1,3 bis-phosphoglyceric acid. And that is the 6th step here. In the 6th step, what is happening, students? Oxidation is taking place. Oxidation. This is undergoing oxidation. But how are we getting the NADH? NADH is produced as a result of reduction. This is how you understand glycolysis, students. This is how you understand glycolysis, not by by-hearting it. Okay. Now understand all of you. Payoff stage, right? Payoff stage. Now what do we have, students? 6th step is done now. 6th step is done. 6th step, 1,3 bis-phosphoglyceric acid. Now students, tell me all of you, where is all the energy stored? Remind me one more time. Where is all the energy stored? In the starting of the class, I told you all the energy is stored in the bonds. What if I break the bond now? What if I break the bond? If I break the bond, energy will be liberated. That is what is happening right now. So if this is 1,3 bis-phosphoglyceric acid, if I break the bond open, energy is released, phosphate is released. Let's do that. And we can use the same energy, and if you take this phosphate and you add it to a ADP, what do you get? Take the energy, take the phosphate, take the energy and add it to the ADP, what do you get? ATP. So in the 7th step of glycolysis, finally we are getting not 1 ATP but 2 ATP. Why? Because everything is into 2. So 1,3 bisphosphate will give you 1 ATP. Since we have 2 molecules of it, we are getting 2 ATP here. 2 ATP we are getting here. Crazy? Yes. Now understand, students. In the 8th step, what is happening? Again, isomerization is happening, nothing. So what is happening is 3-phosphoglyceric acid, what we have right now. In the 8th step, so in the 7th step, 3-phosphoglyceric acid, right? This phosphate is now sent to the 2nd position. Isomerization, right? Same chemical formula, structurally they are different. So 2-phosphoglyceric acid to 3 to 2. So 2-phosphoglyceric acid. So what is happening in the 8th step? Isomerization. Isomer formation is happening in the 8th step. Done. Now students, what is happening in the 9th step? In the ninth step, there is a loss of water. Ninth step, there is a loss of water. That is, 2-phosphoglyceric acid is losing water to get phosphoenol pyruvate, PEP. Phosphoenol pyruvate. C4 cycle. Same thing. Phosphoenol pyruvate. Now again in the 10th step, what is happening? This high energy molecule, bond is broken, phosphate is released, energy is released. This phosphate is added to a ADP and we get one more ATP. In the 10th step, we get one more ATP. And that is the 10th step process of glycolysis. Now let me tell you students, how can they ask the questions and which type of questions also. That is my job to tell you. Okay, this is the entire reaction you can see here. Let me write it one more time. First step, ATP is consumed. Second step is isomerization. Third step, again ATP is consumed. It is consumed. Fourth step, what is happening? 6 carbon to 2 3 carbon. Fifth step, isomerization. Sixth step, oxidation is happening. In the seventh step, again ATP is produced. Eighth step, again isomerization is happening. Ninth step, water is being removed. And the 10th step, again ATP is being formed. You need to know exactly each step by step what is happening. If I ask you which step ATP is formed, 7th and 10th. Which step ATP is consumed, 1 and 3. Which step oxidation takes place, 6th step. Which step water is removed, 9th step. You don't have to just remember the steps, you also need to know what is the exact reaction in every single step. That is more important. If I ask you 6th step, what is happening? You should be able to tell me, sir, glyceraldehyde 3-phosphate is getting converted to, remember oxidation, 1 phosphate was added extra. So 1,3, yes. So 1,3 bisphosphoglyceric acid. What is happening in the 7th step? ATP is produced. So if ATP has to be produced in 7th step, one bond has to be broken. It has to be broken. So 1 phosphate has been removed. It's been removed. So only 3 is there. 3 phosphoglyceric acid. In the 10th step, again ATP is produced. PEP to pyruvate. 10th step. So you need to know every single step in glycolysis by understanding it visually. Only when you understand the concept, you don't have to by heart any of it anymore. Nice, no? How many of you enjoyed? Tell me, students. How many of you enjoyed the entire? Now, what is the net gain here? Very simple. Net gain is very simple. Child's play. Okay. In preparatory stage, minus 2 ATP. 2 ATP is consumed in the first and third step. Okay. Now, in payoff stage, we have 1 NADH. Or 2 students, tell me. 1 or 2 NADH? 2 NADH. Which step? 6th step. Okay. 2 NADH. And I told you 1 NADH is giving you 3 ATP. So into 3. 1 NADH will give you 3 ATPs. 1 NADH, 3 ATP. So into 3. 3 2s are, tell me students, 6 ATP. And we also get 2 ATPs in general. 2 ATPs from one side. One more 2 ATP in the 7th step. 2 ATPs in the 10th step. Tell me students, what is the gross energy produced? Gross energy produced here is 6 plus 2 plus 2, 6, 7, 8, 9, 10. Gross is 10 ATP. Gross is 10 ATP. Now, but we used minus 2 ATP in the preparatory. So, 10 minus 2 is net ATP. So, net ATP gain is 8 ATP gain. Net ATP gain is the 8 ATP gain here. This is the net. This is the net ATP gain here. Done? 10 students. That is the net ATP gain in the case of glycolysis. Now you might ask me which type of question I can expect here. Very simple. Let me tell you. First question on the board, all of you. If you know the answer, tell me the answer. Okay. Which step ATP is consumed? Tell me. Consumption 1 and 3. 1 and 3. Which step ATP is produced? Seventh and 10th. Or they can ask you, which is, let's say, rate determining step. Right? They can ask you, which is the rate determining step? Rate determining step. You will say third step. Which steps are irreversible? Extra question. This is an extra question. Sometimes they can ask you, which are irreversible steps. Irreversible steps. You will tell me, sir, it is 1, 3 and 10. 10th step. Or they can ask you, which step oxidation takes place? Oxidation in which step? Oxidation takes place in the 6th step. In the 6th step, where PgAl gets converted to, right, 1,3 bis phosphoglyceric acid. You have to remember it like that also. Okay. So, the 6th question. What is EMP pathway? What is EMP pathway? They can ask you. 7th step. Which step water is removed? Which step water is removed? Tell me 9th step. 9th step. So, the question can keep on going on. I can ask you one more question. Does it happen in aerobes or anaerobes? Tell me, students. Does it happen in aerobes and anaerobes? Tell me in the chat. It takes place in the case of both. In the case of both. Or they can ask you enzyme also. So remember, students, first enzyme and last enzyme, that is the most important. The first enzyme is called as hexokinase. The first enzyme is called as hexokinase, and the last enzyme in the 10th step is called as pyruvate kinase. Pyruvate kinase. Okay. So such question can be asked in examination from glycolysis. Apart from that, they can ask you, what is N-type process called as? This N-type process is called as substrate level phosphorylation. We are using a substrate to produce energy. Using a substrate to produce energy. The substrate here is glucose. The glucose is being used to produce energy. Okay? Clear, all of you? Here's the question on the board. Let's see who can answer. Question on the board for all of you. Question on the board for all of you, students. Tell me the question on the board, students. Question on the board for all of you. In plants, sucrose is converted. Sucrose is converted into two molecules of monosaccharides due to the action of invertase, and then both enter glycolic pathway. Identify the monosaccharides. I already told you. The two monosaccharides which can enter, is glucose and fructose. Glucose and fructose, because sucrose can be divided into glucose and fructose, right? Glucose and fructose. Next question. In glycolysis, the conversion of 1,3 bis phosphoglyceric acid into 3-phosphoglyceric acid. 1,3 to 3. Students, if you know the concept, forget if you don't know the steps also. If you know the concept here, you can say there is 1 phosphate to 2 phosphate as there, and here there is only 1 phosphate. So can I tell 1 phosphate was removed? If 1 phosphate is being removed, it means it was converted to ATP, using ATP. Yes or no? So can I tell, even if you don't know the steps, if you forget the steps also, But if you learned the concept with me, you can tell me, sir, it is an energy yielding process. Energy is produced here. See, that's how you learn the concept. Okay. Next question. ATP is used in two steps. It is used in two steps, 1 and 3. All of you know, right? First step is used to convert glucose to glucose phosphate. Done. This is the first step. Second, ATP is used to convert fructose phosphate into fructose 1,6. Yes, in the third step it is used. True? True. Both A and true, and it is the correct explanation. Yes, correct explanation. Correct explanation. Next question. Conversion of glucose into glucose 6 phosphate. The first irreversible. See, I told you 1, 3 and 10 are 3 irreversible steps. First irreversible reaction, glycolysis, is catalyzed by. I told you already, you need to know the first enzyme and last enzyme. The first enzyme here is hexokinase. Hexokinase. Next question. Sucrose is converted to glucose and fructose by the enzyme. Again, NEET PYQ. Invertase. Invertase. Next question. Glycolysis is found in cytoplasm of virtually all types of aerobic and anaerobic cells. In this process, glucose is converted to, basic question, all of you know, glucose is converted to pyruvic acid till the very end. It's a AIMS 2002 question, AIMS question. Okay. Next question to all of you. In which of the following reactions of glycolysis, a molecule of water is removed from the substrate? I told you, water is removed in the 9th step. Yes or no? In 9th step, where we get PEP, phosphoenol pyruvate. Yes or no? So option number C. Option number C. Phosphoenol pyruvate. 9th step, water is being removed. Done. So do you realize when you learn the step by step and you integrate it and learn it, all of these questions are so easy to do. So easy. It may be AIMS level, NEET level or AIPMT level. Here's the question. Find the odd one out statement with respect to plant respiration. Glucose is derived from the end result of photosynthesis of sucrose. Yes. Invertase act on sucrose to release glucose and fructose. Yes. Glucose and sucrose are phosphorated in the presence of hexokinase. No. Why? Because sucrose cannot enter the glycolysis. Sucrose has to be converted to glucose and fructose. Only glucose and fructose can enter the glycolysis, not sucrose. So wrong step, wrong question, basically wrong statement. Okay, students, that is the end of glycolysis. I believe all of you understand glycolysis. All of you can solve the question of glycolysis, and I hope all of you truly understood the glycolysis. If you did, some fire emoji in the chat to know that yes, all of you are following and you're understanding. And with that being said, now we move to the next concept for today, is called as fate of pyruvate. Now, after entire 10 steps of glycolysis, we get the pyruvate. So what exactly happens to this pyruvate now? What is the next life cycle of this pyruvate? Can we start? Can we start now, students? Can we start? What is the fate of this pyruvate? Like what does it undergo? What is it undergonna go now? Fate of this pyruvate. So before we go to the fate of pyruvate, let's understand what exactly happened till now. We had the glucose, right? We have the glucose. Glucose undergoes a reaction to produce pyruvate. Okay, to produce pyruvate. This entire reaction is called as glycolysis. Students, tell me how many carbon? Glucose is 6 carbon. Pyruvate is 3 carbon. 3 carbon. Okay. Now, one more thing I forgot to tell you here is the entire process of glycolysis. Is said to be partial oxidation. Partial oxidation. You might ask me why? Because students, in partial oxidation, there is energy produced, but there is no CO2 release. So students, did you find anywhere in the entire glycolysis anywhere CO2 was released? Absolutely not. CO2 is not released at all anywhere. So this is called as partial oxidation. No CO2 evolved. Okay. Now what happens with pyruvate? Now this pyruvate right here can undergo 3 different pathways now. It has 3 life options. Okay. The first 2 are in absence of oxygen. The first 2 are in the absence of oxygen. Absence of oxygen. Last one is in the presence of oxygen. Presence of O2. Okay. Now understand all of you. The first one takes place in the case of yeast. Second one takes place in the case of muscle cell. In the case of muscle cell, lack of oxygen. And the third one takes place in the case of mitochondria. In the case of mitochondria. So what exactly is happening? In the case of absence of oxygen, in the case of absence of oxygen and in the case of yeast cell, such a reaction takes place called as where alcohol is produced, ethanol produced, and such a reaction is called as alcoholic fermentation. It is called as alcoholic fermentation. This reaction is called as lactic acid fermentation. Lactic acid fermentation. The last one, which happens in the case of mitochondria in the presence of oxygen, is called as your normal reaction, basically students, which happens in the cell, which is called as oxidation reaction. Now here we end up getting the CO2 plus H2O plus energy is produced here. But in the case of lactic acid fermentation, there is lactic acid plus energy. In the case of alcoholic fermentation, there is ethanol plus CO2 plus energy. So this is the entire fate of glycolysis. Entire fate of pyruvate, sorry. Entire fate of pyruvate. Pyruvate can undergo 3 different steps, which we will be studying in today's class only. Okay, today's class. Now the first one is called as anaerobic respiration. Here we have something called as lactic acid fermentation and alcoholic fermentation. Now again, how do you want to learn it, students? How do you want to learn it? Do you want to simply by heart it, which does not take more than 5 minutes, or do you want to truly understand it? It is up to you, right? I can make you by heart this in 5 minutes, which doesn't take a lot of time, rather than it takes more time for you. For me, it only takes 5 minutes to make you by heart. It takes more time for you to by heart it later on, sit and memorize it. Or do you want me to make you understand the concept of the fermentation also, like how I did for glycolysis? Let's understand. Okay. Now, the first one is called as lactic acid fermentation. Lactic acid. All of you know it takes place in the case of muscle cells or in certain bacteria, lactic acid bacteria, LAB, right? Which is used in curd making and everything, LAB, right? So what exactly happens here? Understand all of you. Okay. Now here, pyruvate is called as the first electron acceptor. First electron acceptor right here. So what do you need to by heart? I'll tell you first of all. What do you need to by heart here? Okay. Now what you need to by heart here is this enzyme. This you need to by heart. Lactate dehydrogenase. But what you need to understand here is very simple. Okay. Remember students, this NADH which was produced in the glycolysis, this NADH in the 6th step, it is produced in glycolysis. This NADH undergoes something called as reoxidation. What do I mean by reoxidation? Technically oxidation only. I call it reoxidation because it underwent oxidation and it was produced that time in 6th step of glycolysis. Now it undergoes reoxidation. Now tell me students, what is the meaning of reoxidation? Oxidation basically means loss of electrons or loss of protons. They're released. They are being released. So that is why NADH becomes NAD plus. Loss of electrons and loss of protons, it is lost. Now these electrons and these protons are taken up by pyruvate. So that is why I wrote as first electron acceptor. So this NADH undergoes reoxidation and all that electrons and protons are picked up by pyruvate, first electron acceptor. Now you tell me students, what do you call gain of electrons and gain of protons? Gain of electrons and a gain of proton is called as what, students? It is called as reduction. So this pyruvate undergoes reduction to produce lactate. 3 carbon, no change in carbon. There is usage of one NADH in the presence of an enzyme called as lactate dehydrogenase. Lactate dehydrogenase. I hope all of you can remember this now. Very simple. Reoxidation, electrons and protons are picked up by pyruvate. Since it is taking electrons and protons, it is undergoing reduction to produce lactate. Same concept also applies here. But in the case of alcoholic fermentation, you need to understand here is that acetaldehyde is the first electron acceptor here. Why? Because pyruvate here directly doesn't become ethanol. No. All of you remember, in the case of alcoholic fermentation, there is a release of CO2. Yes, there is a release of CO2. That's how dosa batter becomes, that's how cheese, we get holes, and that is how the curd or the bread becomes bigger and bigger. Bread becomes bigger and bigger. Bread. Okay. So how do we get this CO2? So what exactly happens here is the pyruvate undergoes a reaction to release one CO2. Can you see here? In the presence of enzyme called as pyruvate decarboxylase. So one CO2 is released outside. 3 carbon, 2 carbon. Makes sense? Makes sense. Pyruvate decarboxylase, removal of one carboxyl group. Right? Pyruvate decarboxylase. That's how we get acetaldehyde. Now understand very carefully, all of you, very carefully. Same thing happens here. This NADH undergoes reoxidation. When it undergoes reoxidation, electrons and protons are removed. They are removed. Electrons and protons are removed. Now these electrons and protons are taken up by the first electron acceptor, that is acetaldehyde here. Acetaldehyde. And this acetaldehyde, when it is taking electrons, taking protons, it is undergoing a process called as reduction. In the presence of enzyme alcohol dehydrogenase to get ethanol. So here we had, I think this you can remember easily, pyruvate decarboxylase, alcohol dehydrogenase. Similarly here we had lactate dehydrogenase. Here we have alcohol dehydrogenase enzyme, and that's how we get ethanol, also type of alcohol. And that is the entire process of anaerobic respiration, where we have glucose, pyruvic acid, lactic acid, ethanol. NADH is getting reoxidized to NAD+. Now it is getting reoxidized. Now what exactly happens here is this NAD+ is going back to the glycolysis, and that is how NADS replenishment also happens, to make sure NAD+ doesn't go out of business. Okay, so this NAD+ again is sent back here, undergoes oxidation to produce, and this here it undergoes reduction, produce NADH, coming here, used here. Now you might ask me, sir, in this anaerobic respiration, how does the energy being produced? How much of energy is produced? Think again, think again. Now till here it is same, right? Till here, till pyruvic acid, it is normal glycolysis. So again in glycolysis, there we have preparatory stage. Preparatory stage we have -2 ATP, -2 ATP. In production of pyruvic acid, we have in payoff stage we have 2 NADH. So we get 6 ATP here. And we also have 4 ATP being used, 4 ATP being produced normally. 2 ATP in 7th step, 2 ATP in 10th step. Like, so here this is 4 ATP. So in normal glycolysis, we have gross is 10, 10 minus 2 is 8 ATP is normal glycolysis, 8 ATP. But in the case of alcoholic fermentation and in the case of lactic acid fermentation, this NADH which is produced here is technically being consumed here. So can I right now remove this part from the calculation since it is being used already? So what do we have right now? 4 minus 2 is 2 ATP. So the net ATP. So the net ATP in the case of alcoholic fermentation is 2 ATP. 4 minus 2 is 2 ATP. So do you realize that in the case of anaerobic respiration, I told you in the starting, very less ATP is produced. Hence the proof. Aerobic respiration, more ATP, anaerobic respiration, less ATP. It is actually less than 7% of energy being produced. Less energy is being produced here. Okay. Yes. So this is all technical thing, right? All is technical. Students, if you're able to follow, quickly smash the like button, right? So if you're watching my lecture for the first time, you'll understand how technical we go to this chapter. I can simply make you by heart this, right? By heart this, but that is not how you learn this chapter. You learn this chapter with the help of concepts, and that's how we are dealing here. Okay. Now, a small question on the board. Choose the wrong statement. Okay. CO2 is harmful molecule and released during catabolic reactions. Respiration will always require O2. Breathing is part of respiration. All of the above. See, correct, students. Correct. CO2 is harmful molecule. That's why we remove it out. Okay. Catabolic reaction. But students, do you think respiration will always require O2? No. Because we just now learned in certain type of respiration, lactic acid, alcoholic respiration, there is no requirement of O2. No requirement of O2. Okay. Next question. What amount of energy is yielded from glucose during lactic acid fermentation? Less than 7%. Less than 7%. It's a NCERT line. Okay. So with that, we finished anaerobic respiration, done and dusted. And I believe it took us almost 1 hour. Almost an hour. Yes or no, all of you? It took us almost an hour to finish the entire anaerobic respiration, glycolysis and everything. Now students, now we go to the aerobic respiration. Typical aerobic respiration, carbon dioxide plus water is released, energy is released here. Now you might ask me, where does it take place? Till now, all the reactions were taking place in the case of cytoplasm, glycolysis. Now students, this NADH produced in glycolysis, as well as pyruvate produced in glycolysis, now they enter inside the mitochondria. So glycolysis, glucose, pyruvate enters inside the mitochondria. Okay. Now inside mitochondria, once pyruvate is entering mitochondria, it undergoes now 3 main steps again. One is called as link reaction, also called as oxide decarboxylation. Second is called as Krebs cycle, and the third one is called as ETC, also called as oxidative phosphorylation. That is addition of phosphate using oxygen. Okay. Crazy next steps are. So this is the entire flow for the next 1.5 hours. Okay. Oxide decarboxylation, Krebs cycle, ETC. Where does it take place? Mitochondrial matrix. Mitochondrial matrix cristae. What is cristae? Inner mitochondrial membrane. Let me show you. Okay. Let me show you, students. If this is a typical mitochondria, students, all of you can see, this is a typical mitochondria, right? In this typical mitochondria, if you look around, it has an outer membrane. I believe all of you can see. Oops. All of you can see students, let me tell you. This is a typical mitochondria. In the typical mitochondria, it has a outer membrane and inner membrane. Outer membrane, inner membrane. Between the outer and inner membrane, there is something called as pericentrial intermembrane space. Now, if you look at the inner membrane, it is undergoing a folding that is called as crista. It has own DNA and 70S ribosome. Okay, 70S ribosome. Now, if you look here, this entire part is called as matrix. Now, within the matrix, that is where the link reaction as well as Krebs cycle takes place. Now, what about ETC? ETC takes place in the case of the entire inner membrane. Do you see this inner membrane? So, in this inner membrane space, that is where the ETC takes place. ETC. Okay. Clear, all of you? Clear? Can we focus now? Back to the class. Cristae. Okay. Aerobic reaction means it needs oxygen. Anaerobic means it does not require oxygen. Simple as that. All of you drink some water. Quickly drink some water. Yes. Now, students, quickly smash the like button right now, all of you. What is my name? My name is Indaka. Check my name in the thumbnail. Baswa, you can just, here is my name. My Telegram channel also. Okay. Now, I want everyone to understand what exactly is oxidative decarboxylation. It is also called as link reaction. Also called as the link reaction, or also it is called as gateway reaction. Now, these reactions are a link between glycolysis and Krebs cycle. Between these two reactions, glycolysis as well as the Krebs cycle, between them is called as the oxidative decarboxylation. Now, what exactly is happening here? Understand, all of you. The first thing is decarboxylation. First step is decarboxylation. Now, from this pyruvate, that is 3 carbon molecule, if I remove this CO2, we end up getting CO, one CO here, and CH3. COCH3. This COCH3 is basically an acetyl group. So, the first step is called as decarboxylation, in which one CO2 is removed from pyruvate. And we end up getting acetyl group. Now understand, all of you, understand. This acetyl group now undergoes oxidation. This acetyl undergoes oxidation now. Oxidation. Okay. When this acetyl group undergoes oxidation, it will produce acetyl-CoA. Now, how acetyl-CoA? See, acetyl. Coenzyme A is added here. So, acetyl-CoA. So, acetyl group from here, CoA from here. Now, the small thing which you need to understand here is, in this entire reaction, this oxidation, when the oxidation takes place, this acetyl group will release electrons and protons. These electrons and protons are now taken up by NAD+. If NAD+ is taking electrons and taking protons, it is undergoing reduction. reduction right here. So what exactly is happening, students? Understand, all of you, this is a very important concept called as oxidative decarboxylation. In oxidative decarboxylation, one molecule of pyruvate undergoes decarboxylation. It means removal of CO2. When CO2 is being removed, it becomes acetyl group. Acetyl group undergoes oxidation to produce acetyl CoA enzyme. When it undergoes oxidation, it releases electrons and protons. That is oxidation. These electrons and protons are taken up by NAD plus. If what do you call gain of electrons and gain of protons? Reduction again. Undergoes reduction to produce NAD plus. NADH. And students, this is called link between glycolysis and Krebs cycle. Here we get an acetyl group, which is a fuel for citric acid cycle. And this acetyl CoA now enters the mitochondrial matrix. Students, any doubt till here? If there is any doubt, quickly ask me. Otherwise, I will be starting Krebs cycle right now. Can we start? One thing which I've driven reaction, I have to show you the enzyme and this thing. See here, students, this is what we learned right now. Here. Pyruvic acid, coenzyme A, NAD+. Pyruvic acid is undergoing first decarboxylation. So CO2 is released. It is undergoing oxidation and addition of CoA, acetyl CoA. And in this entire reaction, one NADH is produced. This entire reaction is enzyme mediated, right? It is enzyme mediated. It means pyruvate dehydrogenase enzyme is here, and Mg2+ is the cofactor. So the enzyme and cofactor both are very important here. Pyruvate dehydrogenase is the enzyme, Mg2+ is the cofactor. So remember this point here. Till now we are done here. Now we are starting something called as tricarboxylic acid, also called as TCA cycle, also called as Krebs cycle, also called as citric acid cycle. Can we start? Now? Can we start? All of you. Yes? That is clear? Can we start? Now students, what if I tell you this Krebs cycle is called Krebs cycle because of a scientist. This is a scientist called as Hans Krebs. He was the one who discovered the Krebs cycle. Now students, what if I tell you, I can teach you the entire Krebs cycle in 1 minute? Do you believe that? What if I tell you I can make you remember the entire Krebs cycle in 1 minute? Can we start? Yes, we can start. For example, students, a small mnemonic for you. The small mnemonic is: officer, can I keep selling sandwiches for money? That is all. That is the Krebs cycle. Oxaloacetate, C4 citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, and malate. Let me show you again. See here, right? Can you see here? See here. Same thing. I'll write one more time here. Officer, can I keep, repeat after me, selling sandwiches for money? O4 oxaloacetate, C4 citrate, I4 isocitrate, K4 alpha ketoglutarate, S4 succinyl coenzyme A, S4 succinate, F4 fumarate, M4 malate. That is the entire Krebs cycle in 1 minute. In 1 minute, the entire Krebs cycle. Now students, let me draw the Krebs cycle also for you. Before we read this Krebs cycle, let me draw it for you one more time. Let me draw the entire Krebs cycle. Tell me students, which is the first step in Krebs cycle? Oxaloacetate. I'll tell you exactly what you need to remember also. Oxaloacetate. Now, oxaloacetate gets converted to what, students? Oxaloacetate gets converted to citrate. Officer, can I? Citrate. Now here I want everyone to understand the carbon number. Oxaloacetate is 4 carbon. Citrate is said to be 6 carbon. So can I tell when 2 carbon is been added here? The 2 carbon is coming from entry. Who's giving the entry here, students? Tell me. The entry is given by acetyl-CoA from the link reaction. And this acetyl-CoA is 2 carbon molecule. 2 carbon molecule. We learned here, right? We learned here. Acetyl. Right? This is the acetyl group. 2 carbon molecule is acetyl-CoA. This acetyl-CoA is entering the Krebs cycle. Here. Entering the Krebs cycle. Oxaloacetate to citrate. To citrate. Okay, to citrate. Now citrate will get converted to isocitrate. Converted to isocitrate. How many carbon, students? Again, carbon number is 6 carbon. 6 carbon. Now students understand, isocitrate will get converted to something called as, officer, can I keep selling? Officer, can I keep alpha ketoglutarate. Alpha ketoglutarate. Alpha ketoglutarate. Right? Alpha ketoglutarate. Now what happens here, students? This is 6 carbon. This is 5 carbon. Now what exactly is happening here is, remember I told you something called as oxidative decarboxylation before. Oxidative decarboxylation, where there's production of NADH and loss of carbon. That is what is happening here. Now in this entire cycle you will learn, this entire oxidative decarboxylation will happen 2 times. The first time takes place here, and with that there is production of one NADH as well as release of one CO2, and that is why 6 carbon to 5 carbon. 6 carbon to 5 carbon. Now alpha ketoglutarate gets converted to succinyl coenzyme A. Succinyl coenzyme A. In this step also there is second oxidative decarboxylation, and there is release of NADH and there is release of one CO2. If there is release of one CO2, students, tell me the carbon number. 5 carbon will become 4 carbon. 5 carbon to 4 carbon is happening. 4 carbon. Now listen to me very carefully. Succinyl coenzyme A. Succinyl coenzyme A gets converted to succinate. To succinate. Succinate. In this step, there is a substrate level phosphorylation. Substrate level, and there is a production of GTP. GTP is being produced here. GTP. Now this succinate now gets converted to fumarate. In this step again, there is a release of FADH. There is a release of FADH. Okay, FADH. Now understand, last step. Now fumarate to malate. In this step, one water is being added. Now, finally, malate will get converted back to oxaloacetate, and in this step again, the last step, there is production of one NADH. NADH. That is carbon number 4 carbon, 4 carbon, 4 carbon, 4 carbon, and 4 carbon. Plus 2, 6, 6, 5, 4, 4, 4, 4. everything is maintained as such. Everything is maintained as such. Now do you see here, students? Citrate is becoming isocitrate with addition of an acetyl-CoA. 2 carbon plus 4 carbon, 6 carbon. Now here, students, understand, only these two steps is oxidative decarboxylation. This FADH and this NADH are normal oxidation. It means that there is no release of CO2. CO2 is released only during these two steps. These two steps here. One is from isocitrate to alpha keto to succinyl coenzyme A. In these two steps, NADH is being released. Produced also, CO2 is being released. Okay. Now I can ask you a question. In which step, in one cycle, how many CO2 is being released? You will tell me 2 times. 2 times it is being released. Okay. Now students, let me go one by one each step. What exactly happens? Okay. This is the first step. Oxaloacetate, acetyl-CoA, 4 carbon, 2 carbon, addition, citrate, 6 carbon. Now what? Citrate, we get citrate. That is this step, first step. What happens second step? Isomerization. Citrate becomes isocitrate. Simple isomerization, no change. Okay. Now what happens in this step right here? This is the first oxidation decarboxylation, first OD. First OD, this is the second OD. Oxidative decarboxylation. Now what exactly is happening in oxidative decarboxylation? Understand. This is the entire oxidative decarboxylation. Now here what is happening here is, where is the NADH? Isocitrate here, this is undergoing oxidation. When isocitrate undergoes oxidation, it releases electrons and protons. Okay. It is also undergoing decarboxylation. So CO2 is being removed. These electrons and protons are picked up by NAD plus, and NAD plus is undergoing reduction. Can you see here? NAD plus is taking electrons and taking protons. It is becoming NADH. So this is called as oxidative decarboxylation. It happens 2 times only. First time in isocitrate to alpha keto, removal of CO2, first thing. Undergoing oxidation, electron protons are taken up by NAD plus and NADH, and that is how in this step here we get one NADH and one CO2. Same thing happens in the second step also. Undergoes oxidation, electrons and protons are taken up by NAD plus, undergoes reduction to NADH, removal of one CO2. So second oxidative decarboxylation happens here. Now succinyl coenzyme succinate, one GTP is produced. That is, this GTP is the only step in which substrate level phosphorylation happens. Only where substrate level phosphorylation is happening. Here, both the places, oxidative decarboxylation. Here, normal oxidation. Okay, that's how we get the entire fumarate to malate. When FADH, normal oxidation takes place, and FAD is getting reduced to FADH. Same thing, normal oxidation. NAD plus is getting reduced to NADH, and that is the entire step. What is being added, and that's how we get the entire Krebs cycle. Now here I want everyone to remember in which step OD is happening, first and second OD. In which step GDP is produced, the only substrate level phosphorylation. In which step other two oxidations happening. In which step water is added, and also the carbon number, like 4 carbon, 4 carbon, 2 carbon, 6 carbon, 6 carbon, 5 carbon, 4 carbon, 4, 4, 4, 4 again. So you need to remember the carbon number and the OD also. Clear, all of you? Clear? Yes. Now, the same trick which I told you. Officer, can I keep selling sandwiches for money? what each one of them signifies. Okay. Done, students? Done? Now, students, this is a simple question here. Identify the step in tricarboxylic acid cycle which does not involve oxidation of substrate. In which step oxidation is not happening at all. See, I told you here, oxidation happening here, oxidation is happening. But succinyl coenzyme to succinate, this is not oxidation. This is basically substrate level phosphorylation, substrate level, right? Substrate level. So what is the answer? Succinyl coenzyme A to succinate. That is substrate level phosphorylation, substrate level. Okay. How many times decarboxylation occurs during TCA cycle? Little incomplete, but with acetyl-CoA, with one acetyl-CoA, right? Or for one pyruvate. For one pyruvate, it is happening twice. For happening, it is twice. It is happening. Now, what if I change the question into for one glucose? This will become 4 times. 4 times. 4 times. For one glucose, because one glucose will give you 2 pyruvate. 2 pyruvate means the cycle has to go 2 times. 2 acetyl-CoA. So 4 times it becomes. 4 times CO2 is being released. Okay. Decarboxylation. Now, the 5 carbon compound, told you already, alpha keto, 5 carbon, simple question. Simple question. Number of substrate level phosphorylation in 1 ton of citric acid cycle. Tell me, students. Number of substrate level. Substrate level was only 1. Remember, GTP formation, only 1. Only 1. Okay. Now, students, with that, Krebs cycle is also over. With that, Krebs cycle is also over, which was supposed to be difficult. It is not difficult at all now anymore. Okay. Now we go to the next concept, that is the fate of NADH, FADH. See, it's all good. It is all good to make, right? It is all good to make NADH. It is all good to make FADH. But what is the use if you can't use it? It has to be used somewhere, right? Okay. After this, don't worry. It's a more 30 minutes. The class one is over. Okay. It won't take long. Now, what is the fate of this NADH and FADH? Like, where can we use the NADH and FADH? Right. That is the main issue right now. Because in Krebs cycle, we can only form, but ATP is not formed anymore. Now, the next thing which you need to understand here is this NADH and FADH is now sent to the inner mitochondrial membrane. Okay. And that is where oxidative phosphorylation happens. The meaning of phosphorylation is addition of phosphate, ATP formation, but with the help of oxygen, with the involvement of oxygen. And this happens in the inner mitochondrial membrane. This is the inner mitochondrial membrane. This is the matrix where we had the Krebs cycle, and this is where the entire reaction takes place. What is NAD? What is FAD? All of you know, right? They are basically the electron carriers. They are electron carriers and cofactors. NAD is nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. Okay. Now, students, I'll give you exactly 30 seconds. I want to write down this. There's something called as 5 different protein complexes and 2 mobile carriers. Protein complex 1, 2, 3, 4, 5, as well as ubiquinone and cytochrome C. These 2 are called as mobile carriers. These mobile carriers can take up electrons and move around, basically, or undergo changes also. And I want everyone to draw this. because we will fill up this entire space. Once you draw this, we will try to fill up this entire space will be filled today. Can we start, students? Can we start? Can we start the filling process now? Let's fill this entire. Let's fill it up. First of all, this is matrix where the Krebs cycle is taking place, yes or no? This is the matrix where the Krebs cycle is taking place. Change the color to black. Okay, this is the matrix where the Krebs cycle is taking place. Krebs cycle is taking place. This is the inner membrane. This is the intermembrane space between outer and inner membrane space. Now, students, I'm teaching you ETC now. I want everyone to focus very well right now. If you go wrong anyway today here, you will never understand ETC again. Very difficult to understand ETC. So, maximum attention from now onwards. Maximum attention. Okay. Now, this is called as complex 1. Then we have complex 3, complex 4, right? So, complex 2. Complex 2, complex 4 and complex 5. These are 5 different complexes involved here. 5 different complexes. Okay. Complex 1, all of you know, is called as NADH dehydrogenase. Complex 2, succinate dehydrogenase or FADH dehydrogenase. It can be called FADH2 dehydrogenase. Complex 3 is called as cytochrome BC1 complex. Complex 4 is called as cytochrome oxidase. Complex 5 is ATP synthase. Here is the entire complexes. It is present in inner membrane space. Okay, inner membrane, mitochondrial inner membrane. Now, let's fill it up. Let's fill it up. Here we have FMN, flammine mononucleotide, as well as certain FES, iron sulfur centers. These are basically the electron carriers present inside in complex 1. In the case of complex 3, we have cytochrome C and cytochrome B and iron sulfur centers. Iron sulfur centers. Complex 2 is FAD, right? FAD is dehydrogenase, right? Or succinate dehydrogenase. Here we have FAD, one of the electron carriers. And apart from that, we also have iron sulfur centers. Very simple to understand. Sulfur centers. Complex 4 has mainly copper centers. That is the distinguishing feature here in complex 4 is the copper centers in the oxidase. Okay. Cytochrome A, copper center A, copper center B. These two are the main copper centers. Let me change the color for this one. A3 and cytochrome A1. These are two different types of sulfur centers. This is F0, this is F1. Rotating and bang. Okay. If this is done, students, if this is done, now let's start the entire process of photosynthesis, the entire process of ETC. What exactly happens one by one? Now, follow through. Which color? Let's take dark blue. Okay, very simple to mark here also. Can't be visible. Which color should I take? Let's take red. Now, what happens here is from Krebs cycle, all the NADH is landing on the complex 1. Here we have NADH. Now, once NADH lands here, it will donate the electrons and protons in here, and this will become NAD plus. And when this happens, all the protons are sent here. NADH2 and 2H plus is sent here from here. And when this electrons are being passed, with the entire electron carriers, some amount of energy is produced, energy is released in the entire matrix, in the entire complex. Energy is produced. Using that energy of the entire complex, when electrons are moving, I'll repeat again. When electrons are moving in the entire electron carriers, energy is produced. Using that energy, this entire complex will also shoot out two more protons from the matrix. Two more protons from the matrix. So in total of 4H plus being sent from the entire complex one, 2H plus. Now what about the fate of electrons? The electrons are sent here. 2 electrons are sent here. The 2 electrons here are picked up by your electron mobile carrier called as UQ. Okay. Now this 2 electrons are given to UQ. When electrons are given to UQ, this UQ undergoes reduction to become UQH2. Ubiquinone becomes ubiquinol, UQH2. Now again, this UQH2 will lose the electrons and again it will become UQ eventually. And this cycle keeps on going. Cycle keeps on going. Now eventually from here, the electrons are sent to the cytochrome C, complex 3. Okay. But only 1 electron is sent here. Only 1 electron is sent. From 2 electrons, only 1 electron is sent. You might ask me, sir, why only 1 electron? Because 1 electron is used to maintain the cycle. Now understand small concept here. This particular UQ will also bring in one 2H plus from the matrix into the 2H plus from the matrix pulled here. So that's why UQ become UQH2. 2H plus being pulled here. Now when this 2H plus is pulled here, it becomes UQH2. UQH2, when it becomes again back to UQ, this 2H plus is sent here. Is pulling from here, undergoing cycling using the energy of electrons, and 2H plus and 1 electron is sent to the complex 3. Okay. Now complex 2, FADH dehydrogenase. Right. Complex 2, FADH dehydrogenase or succinate dehydrogenase. It is the enzyme which is also involved in Krebs cycle. Remember that. Now, this particular enzyme, students. This particular enzyme is inner membrane. Okay, it has moved up, students. This is inner membrane. Okay, it cannot send the electrons directly into the intermembrane because this complex 3 is present in inner membrane. It cannot move up and down. Okay, so what happens here is one FADH commences here, FADH or FADH2. This gets oxidized to FAD plus, and when this is happening, all the electrons and protons sent here undergoes oxidation, right? Electrons and protons sent here. Now these electrons are again given to mobile carrier called as UQ. Okay, electrons are given to UQ. Now UQ again gets converted to UQH2. Now you might ask me, sir, how did the H2 come again? Did it pull this H2 again from the matrix? No, not this time. This H2 right now was again provided by this H2 here. Understood? Now you might ask me, sir, why can't we just take this H2 and send it here directly out to the intermembrane? Not possible. Why? Because this protein, complex 2, is present only in the inner membrane, not like other complexes. It is present only in inner membrane. Okay, it cannot send here. So what is happening here is 2 electrons are being circulated here. 2 electrons sent here to 2 electrons. Now from here again, just like how we did here, 1 electron is sent to the complex 3. 1 electron is sent to the complex 3. And at the same time, this entire 2H plus given from here also goes here, 2H plus. Now you tell me, students, in total of how many electrons and how many protons are going to complex 3? Now tell me in the chat, to the complex 3. Tell me. In total of one electron, 2 electrons, 2H plus, 2H plus. So 4H plus. So this 4H plus will be sent out to the intermembrane space, and these 2 electrons here is sent to a mobile carrier called as cytochrome C. Cytochrome C. Electron sent here. Now this cytochrome C will travel from here and it will reach here eventually. It will reach here. It will reach here. Okay. It will reach here. Okay. Now it is reaching here and it is transporting all the electrons. 2 electrons. Now, here comes the final boss of this story. This entire journey of respiration, all the way from the glycolysis to Krebs cycle, link reaction, and etc. I told you this entire thing happens in aerobic respiration. But did you see anywhere till now, oxygen, students, tell me, anywhere this entire process of etc. or entire process of Krebs cycle or entire process of glycolysis, did you anywhere see oxygen involvement? No. So it is said that the role of oxygen in aerobic respiration is only and only at the very end. And this is the entire role of oxygen, students. The entire role of oxygen is to pick up the electrons in the end. That is all. So what happens here is half O2, different color I'll use, not visible, no? Half O2 plus 2H plus. Half O2 plus 2H plus gets converted to water molecule. And that's how we get water molecule as a byproduct in this entire reaction. Okay, and this is the entire glycolysis. This is the entire etc. Okay, now what is happening here? Once the electrons are being sent here, protons are being sent here. Now what happens is this 2H plus also, when these electrons are being passed, when these electrons are being passed from here to here, it is generating energy, and with the help of that energy again from intermembrane, from the matrix to the intermembrane space, 2H plus is sent here. 2H plus is sent here. So in total of how many H plus do we have right now? 4H plus here, 4H plus here, and 2H plus here. So that makes up total of 10H plus in the intermembrane space. In the intermembrane space, total 10H plus. Out of this 10H plus here, every single time 4H plus goes in. Every single time 4H plus goes in, 3H plus is used to rotate the ring, 1H plus is used to bring the phosphate, and every single time that happens, 1 ADP gets converted to 1 ATP. 4H plus 1 ATP we get. 1 ATP we get. And that's how entire body, in our body, this is happening right now. And it doesn't look like this so well. Let me show you how it looks. Before that, I'll give you a small quick revision of this entire thing. Follow me through. Okay. Super amazing, right? Do you realize this all is happening in your body? Okay. So first, matrix, intermembrane, inner membrane, intermembrane. Complex 1, complex 3, complex 2, complex 4, complex 5. Complex 1, NADH dehydrogenase, NADH is coming and it is giving 2H plus here from here. Okay, 2H plus. When electrons are being passed through the different types of electron carriers, energy is produced. Using that energy produced when the electrons are being transported, 2H plus again is transported here. So totally 4H plus. Now, same thing happens here. FADH gets converted to FAD plus, electrons and protons are transported here. In both the cases, electrons are sent to an intermediate electron carrier called as ubiquinone. This ubiquinone picks up the electrons, 2 electrons from here, 2 electrons from here, and gets reduced to UQH2. It is becoming UQH2 because of it is taking 2H plus from the matrix here. It is becoming UQH2 here because it is taking the 2H plus from here. Different locations picking up electrons and protons. Okay. Now out of these 2 electrons, only 1 electron is sent here, 1 electron is sent here. Why? Because 1 electron is used to maintain the entire cycle. 2H plus from here is sent here, 2H plus. So in total, complex 3 gets 2 plus 2, 4H plus. 1 plus 1, 2 electrons. Now these 2 electrons has to go from here to here. Possible? Yes, possible. Because of mobile carrier cytochrome C transporting here. From here again, electron goes up, goes up, goes up. Final electron acceptor is half O2. I didn't write O2 only, yeah. Of oxygen. Okay, half O2 plus H plus gives you water. Okay, electrons are going, and when these electrons are moving, energy is used to send back 2H plus into the matrix. So total 10H plus here, 4 plus 4 plus 2, 10H plus, and it's 10H plus every single time out of 10. 4H plus is sent here to complex 5 ATP synthase, 1 ATP is produced. So 4H plus 1 ATP, 4H plus 1 ATP is produced. Okay, 1 ATP is produced here. Now, how does it look? How does it look like? Like, students, see this. This is the entire mitochondria. In the inner membrane, of mitochondria, this is the inner membrane. Can you see inner membrane? Lipid bilayer, right? Lipid bilayer is going to be there here also. Okay. Can all of you see lipid bilayer? This is the matrix on the top. I told you intermembrane. Middle part is inner membrane. Same like the diagram. Matrix, intermembrane, inner membrane. Here we have the ATP synthase, that is basically the complex 5, F0, F1, rotating, catalytic inaction. And see, can you see ATP formation happens here? ATP molecule. What is this? These are H plus. As the H plus are going up, ATP is being formed. 4H plus in, 1 ATP molecule on top. 4H plus, yes, technically 4H plus gives 1 ATP. Okay. Now we will show the other complexes. Can you see here? Complex 2, complex 3, complex 1 first, complex 2, complex 3, complex 4. Different types of complexes are working together here. The first complex is the complex 1, NADH dehydrogenase, where the NADH comes in sets. NADH. And this is how it looks. And there are multiple electrons going in and out. Can you see? Electrons are moving in and out. Goes in. What is this coenzyme Q? Coenzyme Q is basically this ubiquinol. Okay. I told you complex 2 cannot send directly. I told you complex 2 cannot send the protons directly. FADH, all the electrons are sent as supposed to be sent. It has to be sent to the ubiquinol only. It is an intermembrane protein. I told you, when the electrons are being transported, they produce a lot of energy. I told you. Yes. See, this is complex 4, this is cytochrome C, mobile carrier is going. Complex 4. If this is the electron carriers everywhere, electrons are being sent here. That's how they pass. Last, oxygen comes in, final electron acceptor, and it becomes water. It becomes into water. Complex 4. H2O formation happens. Water formation takes place. And that's how it looks like. It looks like, see, there are so many of them. Do you see that? On the entire membrane, there are so many complexes, chains going on. One is happening here, one is happening here. Everywhere it takes place, friends. Entire thing. Complex 1, complex 3, complex 2, complex 4, where water formation happens. This is the entire ETC, electron transport chain, electron transport system. Okay, this is the given density diagram. This has to be rotated this way. This is the given vertically. See, matrix, intermembrane, 1, 3, 2, 4, 5. So I've made it straight. Basically for you. Okay. Now here is a small question. If you know the answer, very simple right now. Which is complex 1, complex 2, complex 3, complex 4. Right? Tell me quickly. Complex 1, NADH dehydrogenase. Complex 1 is called as NADH dehydrogenase, done. Complex 2 is called as FADH dehydrogenase. So A is 3, B is 4, answer is 2. Answer is 4. Answer is 4. Cytochrome BC1, complex 5 is complex 4 is cytochrome oxidase. What is the role of NAD plus cellular respiration? Tell me, students, what is the role of NADH, NAD plus? It is an electron carrier. We learned now it is a type of electron carrier. Right? Now, simple question again. Citric acid cycle takes place in the mitochondrial matrix. Glycolysis happens in the case of cytoplasm. So A is 2, B is 1, electron transport chain, inner membrane. C is 4, D is 3, intermembrane. D is 3. So easy question, easy answer. Next question. Which of the following statements are correct about respiration? Energy of oxidation reduction is utilized for phosphorylation. Yes. One is going oxidation, one is going reduction. Okay. 2 is actually wrong. 2 is actually wrong. It's 4. Technically, it's 4 only. 2 cannot give anything. Okay. Oxygen acts as a final electron acceptor. Correct. Photooxidative energy is utilized in production of proton gradient required for phosphorylation. No. Photooxidative photosynthesis, not here. Role of oxygen is limited to the terminal stage of the respiration. Yes. Protons are cross the outer membrane. No. It is supposed to be inner membrane, right? Inner membrane. So ABD. ABD. Create pair. Identify the cytochrome which acts as a mobile carrier. Mobile carrier, cytochrome C. Mobile carrier. Now, students, that was the entire ETC is also over. Electron transport chain system is also over. If you think that was difficult, it just became very easy. Now, if you're watching my lecture for the very first time and you are confused that, sir, how detailed it is, I would recommend all of you to watch it one more time in the 2x speed if possible, because I have gone as slow as possible in discussing every single topic today. So I've gone as slow as possible. So watch the lecture one more time for get a better clarity on this topic. Now let's do entire respiratory balance sheet, students. Entire respiratory balance sheet. Can we start? Can we start? Respiratory balance sheet. Now first of all, let's take glycolysis into account. Let's take glycolysis. What exactly happens in glycolysis? All of you know -2 ATP in preparatory. Then there is NADH. NADH gives 2 molecules of NADH. 2 molecules of NADH into 3, that was 6 ATP. 6 ATP. 4 ATP was produced as such. So in total there was gross is 8 ATP. 10 minus 2 is 8 ATP. Then there is link reaction. Link reaction gives 2 NADH. You might have said only 1 NADH you wrote that time, but this is for 1 glucose. For 1 glucose we are doing right now. Okay. So 2 NADH. So I'll give you 6 ATP again. 1 NADH is 3 ATP. 3 into 2 is 6 ATP. That gives 6 ATP here. Now what about Krebs cycle? In the case Krebs cycle, we have 6 NADH in total. 3 for 1 pyruvate. 6 for entire glucose. 2 from decarboxylation and 1 from last step. So 3 are there in one cycle, but we are doing for 1 glucose. So 2 times it has to go, right? 2 times. So 6 NADH. And we have 2 GTP and 2 FADH. 1 FADH in one cycle, but we are doing for 1 glucose, so 2 FADH. 6 into 3 is how much? Into 3 into 2. Right? Into 2. 1 FADH gives 2 ATP. 6 into 3 is 18 ATP. 18 ATP here. This is 2 ATP here. And 2 is 4 ATP here. That makes up the total. 18 plus 2 is 20, 20 plus 4 is 24 ATP. 24 ATP. So what is the grand total? Grand total is going to be 24 plus 6 is 30, 38. 38 ATP. 38 ATP is the grand total. That is the number of balance sheet in the case of the and for one glucose. For one glucose, we get 38 ATP. But let me tell you, students, it is not the actual. It is not the actual thing. In reality, there is only 30 ATP maximum, 30 or less than that also, because there is something called as, it is not 3 and 2, it is 2.5, 1.5 first of all. And second thing is, when we have to transport the NADH into this NADH, when you have to transport into here, it takes more energy for that also. So reality is different, but NCERT does not tell you the reality. So stick to what is given in NCERT right now. So that's 38 ATP. Okay. Now students, the last topic is called as call for backup. Now what do I mean by call for backup? Like for example, if you have inverter in your house, like for example, light goes. What do? Do you sit in darkness? No. If you have a UPS, you switch on the UPS. UPS is the backup. Similarly, inside your body also there is a backup. Just because carbohydrates are not available at the point, our body will never starve to death. Body is very smart. And immediately it will switch on the backup. Now what is the backup here? The backups are proteins as well as fats. So as soon as carbohydrates are over, our body is switching to fats and proteins for consumption. Okay, that is basically called as the backup. So what happens? There's breakdown of fat and breakdown of proteins. Now let me teach you how does the fat gets broken down. How fats gets broken down, students. There are two ways in which fats can be broken down. First of all, fats are mainly of two types. If you know biomolecules, there are two types of fats. One is called as fatty acid. And other one is called as glycerol. Both of them undergoes oxidation. Here, beta oxidation, to be precise, undergoes oxidation. When it undergoes oxidation, fatty acid produces acetyl-CoA. Students, do you remember acetyl-CoA? Plus energy. Do you remember acetyl-CoA? Acetyl-CoA will enter the Krebs cycle. Krebs cycle will undergo ETC. ETC produces energy. And this glycerol produces DHAP. DHAP, all of you remember? DHAP is part of glycolysis. Gets converted to pyruvate. Pyruvate becomes acetyl-CoA. Acetyl-CoA becomes enters the Krebs cycle. Krebs cycle gets converted to ATC, and that's how the energy is produced. This entire thing goes on. So fats can be used to produce energy when needed. We don't have to rely on carbohydrates only all the time. Just because your favorite biscuit is not available, it doesn't mean you can go for alternative. You can go for alternative. Try a hide and seek one time. Okay. Now, what do you mean burning of fat? Let me tell you, students, if you want to decrease weight in general, right? If you really want to burn the fat in your body, you have to make sure first of all that your carbohydrates intake is very less. It is called as calorie deficit. Now, what does the meaning of calorie deficit? It means that if you don't increase the carbohydrates intake, if you do not decrease the carbohydrates intake, our body will never burn the fat in general. So if you want the body to burn the fat, first decrease the carbohydrates, and then the body will directly attack the fats and break down the fat. Okay, it will actually even go for protein also. That is why bodybuilders eat more protein to substitute that, and bodybuilding also. Okay, so proteins can also be broken down. This mainly happens in the case of animals. Okay, protein gets converted to amino acids. Amino acids like glutamic acid gets converted to alpha ketoglutarate, undergoes Krebs cycle. Aspartic acid undergoes deamination again to produce the oxalic acid, undergoes Krebs cycle, energy production. Same thing can happen. This is a general diagram given in your NCERT also. If you see this diagram here, it says that fats, glycerol and fatty acids, glycerol can undergo DHAP, I told you. Fatty acid can get converted to acetyl-CoA. Amination can undergo pyruvic acid. So these are called as alternative pathways. They're called as alternative pathway. One simple question is given here from NEET 2023. Fatty acids are connected with respiratory pathway through, I told you, acetyl-CoA. Yes, acetyl-CoA. Now in the starting I told you, actually not asked, I told you that this entire process of respiration is catabolic. It is catabolic. But what if I tell you right now, it is also anabolic, or there's a new term called as, it is amphiboic process. Now understand the meaning of amphiboic process. In the normal reaction, what happens? Okay, in respiration pathway, we have a glucose. Glucose gets broken down and produces energy. That energy can be used to make amino acids and fatty acids normally. But whenever the body doesn't have this glucose, what happens? These proteins and fats, we just now saw, right? Fats can also be broken down to produce energy. Proteins can also be broken down to produce energy. Yes, they can be. From simple substance, we can make complex substance. From complex substance, we can make simple substance. Catabolism, anabolism. So it undergoes both. So it is called as amphiboic process. Both can happen here. Both can happen here. Okay. And there's something called as respiration quotient. I've added NCERT student because it is so easy to learn. Respiratory quotient is nothing but it's a ratio between volume of CO2 released by the body to the volume of oxygen consumed. How much of CO2 you're producing to how much of oxygen consuming, depending upon the substrate which you eat. For example, if I'm taking carbohydrates, equal amount of oxygen and CO2 are used. Okay, see here, it's almost 1 for carbohydrates. But when fats are used in respiration, an example is tripalmitin, it is 0.7. That is, we need more oxygen. Can you see? We need more oxygen and we release only less CO2. But when you take out proteins is even more less. When proteins are respiratory substrates, the ratio will be around 0.9 for proteins, 0.7 for fats, and 1 for carbohydrates. And with that, this entire chapter is over. Do you realize, students, in one shot, we finished the entire chapter in one go today. Right? I hope you understand different concepts. I hope you were able to understand with the help of mitochondria and the ETC video which I showed you. And also at the same time, I hope you were able to understand the glycolysis better today. Okay. So students, last day for Christmas sale, I'm telling you again. Jan 1st, Christmas sale gets over. NEET Express batch, very few limited seats left, probably like around 10 seats are left. So if you have not joined the NEET Express, join right now. And if you have not started the mock test, start doing the mock test right now. And so students, let me tell you honestly, I've told you from last year, I'm telling you, at least 30 mock tests you have to do before your examination. And the earlier you start, it is better. So Christmas sale is on. So go check the description box. You'll get all the links for different offers going on. So go check it out right now. And do not forget to let me know in the comment section how was today's class and which is the next chapter you want in 3D. Okay. Thank you so much, students. I will see you in the next lecture. Like the video and let me know in the comment section. That is very important. Comments, students, because before a student watch the lecture, they will check the comments first. If the comment tells that yes, this teacher has taught me very well, then only a student will watch the lecture. Otherwise, students don't watch lecture at all. So let me know in the comment section for your friends also to watch the lecture. Okay. So bye-bye. Take care all of you and have a great day ahead. Let me know in the comment section. Ritu, Sharu, Disha, Jenny, Atulya. Let me know in the comment section. Bye-bye. Take care, all of you.